Sorry, class. Today I got late, and then the session I wanted to start was not working as usual. Really sorry for that. Um, we can, those students who join me now, we can, there's this noise coming from outside now. So I got some messages from our MBBT group. I don't want to display his or her name, but his father got dismissed today because of COVID situation, and it's a very heavy hurt that I felt. I've also got some other messages today that things are not happening into our relatives, so it's a very yeah, very sad situation for all of us that our families, we have to go through this time. Um, so whosoever have joined me today, I really request. I mean, we can't help in this meantime, but before we sleep or we wake up in the morning, just pray for the all the rest of us that who we are and who have our dear and near ones that we have lost. Just pray for everyone's well-being, that everything gets better as it was in 2019. Now we started to have normal life, and we don't see, especially our dears and near ones, going passed away. So just at least we can bring some good thoughts, good energy to the atmosphere. Um, Even a single iota is enough to make um, an ocean. So everyone is a powerful thought. Everyone is a good consciousness. Just try to um, bring good energy to the universe. So let's start with our yesterday session. So we were done yesterday. Uh, we have discussed quite number of microscopy yesterday. From epifluorescent was the last one. Now this is a bit complex, quite big. That is your differential interference contrast microscopy. So it has actually a mixture of both. Your mercury lamp is also there. Your polarizing filters are also there. Your recombining prisms are also there. Even the beam splitter, Wallstein prism, is also there. So four things that have combined. So in this microscope, we are seeing epifluorescence and DIC capabilities coming together. And this DIC microscopy is also known as Normarsky optics. Normarsky optics. So it's a Finnish or Northlandian name, Normaski. So from that, somewhere have might have some work in this. So in this TIC, we resembles the phase contrast. From that, somewhere have um, they are more sensitive, and they gives higher resolutions, and they uses polarizing lenses like the polarizing microscope, and therefore can be quantitative. So the halogen light beam is polarized and split by beam splitter and pass through the specimen. And the split beams are recombined by a second prism in the objective. So whatever the change that we see in the light wave due to the passage through the specimen cause interference in that beam and then produce a difference in the image brightness corresponding to the refractive index of your specimen structure, which gives us a 3D structure, quite high numerical aperture, high contrast, excellent resolution. So everything is excellent, high, much better. 3D images, which we didn't stained our specimens. Even for them, it's pretty good to see the membranes of the cells and to check the change in the refractive index of your sample. For that, it's pretty good. So these characteristics that you're seeing, like all these 3D images, high numerical aperture, high contrast, excellent resolutions for unstained specimens, for refractive index of specimens, we have went through couple of terms here. So 3D image, high numerical aperture, high contrast, excellent resolution, good for your unstained specimens, live mounts, can see membranes within the cells, and detects changes in the refractive index of specimen. So all these things matters a lot in this case. So all these like together makes this DIC an excellent option for working. And somebody was asking me yesterday, sir, which microscope shall we use? So it's depend what kind of images you want, what kind of publications you are going for. If you are going very high end paper, even though you have another from Western blot or from a mouse, from cell culture, from protein data, from DNA data, mRNA data, you have all these different aspect of data, and you want to also show a good resolutions of images. So those microscopes might be not available in your lab, but you can make a collaboration with the other lab in order to find out your results, and those lab people could come to your lab in order to use some equipments that they don't have. So in this way, we used to do collaborations from one lab to another lab. So in this world is all based on your networking. How when it's good that we are good in science, as a scientist, as a science is also good, but beyond that, being a scientist, you also need to have good social what do you say, social etiquettes, no, social power, no, not that word, social skills, yeah, that's the word I was looking for, social skills, uh, in order to attract someone, being a magnetized personality, able to get what you want from someone. And that's a bit being a mean thing, but that's how the world works, because if you want to grow up, if you want to go up, you need to make things done as per your needs, because people might use us, especially that's what I've seen in the science. There will be many people who wants to take advantages of us, but we need to be clever, and the clever one, the smart ones, go to the top place of this hierarchy of academia. So just sharing my own thoughts from the socio-psychology perspective, and which we get to know later on in our lives, that it's good to be hard work, it's good to have scientific mind, it's good to have good IQ, but beyond that, how good is your social skills are, that's also equally important, very important. Being smart, being hard work, and being quick-witted, being humorous, so all these things matters a lot on the long run. Yeah. So I'm teaching you the basic science here today, but these things, the other things that I said, I was not aware when I step into Germany, because I was just till our, this is in India, we are just told that you have 5 semesters, you have to study them and get good grades. So by the end of semester, you get around 8.9 GPA, 8.7 GPA. We seems quite happy, and we give you all your professors keep giving you a lot of assignments in order to get detail of that subjects. So our life is all around these subjects, in order to get the information. That's what the university is. Actually, that's what the university is. That's what the education structure is. That's why we are going for the degree, in order to get the education of one particular stream. But other skills, we tend to forego during this time. We just try to be in the books, and that's it. And that's what I did the mistake during my 4 years of BTech. I was just a bookworm, keep reading things, keep going on. But so when I went into the open world where no one knows me and I want to start my life, so felt some chumps. So I was introvert and not so much extrovert at that time, but things gradually changes with the time. And nonetheless, beyond that, that's keep me going, for my master in the cold winters of Germany was the, oops, sorry. um, so just a bit skip from our today's topic, that is enthusiasm. Its spelling is EN enthusiasm. I think this is the spelling, if I'm right. So if you, according to Greeks, if you separate this ziasm, maybe theo n. This I recently actually read about this enthusiasm. What is the meaning according to the Greeks? According to the Greek language, enthusiasm, n means inside, thu means God, and iasm means vision. That means having a God vision inside yourself. If you have enthusiasm of something in your life for one particular field or one particular interest, that is equivalent to God within. Yeah. So that's how the enthusiasm is such important for our life is. If you keep one enthusiasm for learning in one particular topic, then no one can stop you to reach the heights of Mount Everest. You will achieve it. Oxygen will be less, you will get from somewhere oxygen. The path is very tough to go on top, you will find some way to reach the top. Conditions are harsh, you will be tough at that time. So things keep going on. If things are keep going tough, you will be also getting tough with the situation. So that for that thing, we all need this enthusiasm. So these are the some DIC images, yeah, which is comparison of DIC and fluorescent image here. So on the top, what are you seeing is the DIC image of cricket overall showing oocytes. The oocytes of cricket insect. So where O is your oocyte here, this whole is oocyte, and N is oocyte nuclei. This is the nuclei part, and these B as a total, these small rings, these are called Balbiani bodies. Balbiani bodies. Okay, and also called as mitochondrial clouds. इनको हम we also call them mitochondrial clouds. So this was the DIC image. What you are seeing is, okay, this looks pretty prominent, and what you are seeing is here in this one, um, in the epifluorescent image, the previous microscope that we were seeing. So we are seeing that one. In this, we can see pretty good nucleus, and also these Balbiani bodies shining fluorescently here. So all are these shining, and also these oocytes. and they are stained. Actually, that's the reason for shining for them is that we have stained with the a dye named as rhodamine 123. That's a dye which particularly attached to this Balbiani bodies, and that's why we are able to see these things in it. And these are specifics for the mitochondria. Mainly the mitochondrial part, they are visible. So we can see in these two figures how distinct the image is in the DIC, where we can see proper 3D image and each parts in a very, like, it's like your the other cameras. You know, once you click your picture with your phone, this is phone camera and this is DSLR. So this is DSLR in the modern world, and this is your phone cameras giving you crappy photos. But still, nowadays the phone cameras are now, they're quite, I have to admit, prominent to your DSLR. They're giving uh, how we say this English, takkar dete hai. They're giving takkar, the equivalent to, maybe I don't know how to express that word. आपस में टक्कर दे रहे हैं आजकल फोन कैमरा और DSLR. Both are equivalent to each other. Maybe we can say this. So that was the difference between the both images. So now let's go for confocal laser. That's the I want to speak about. There I had it's one of the precious microscopy, because if we are having 7 to 8 groups are working and 7 to 8 groups are sharing this microscopy, laser microscopy, only the person who have highest skills have is the most accurate and do not do any much mistakes will get the chance to work on them. Only the person who is good in taking care of this microscopy and does not create any mess while doing so, only that person used to get a chance on this. So it's not like everyone just could go for this microscopy. So we need to be very precious because it's expensive. The electron microscope, sorry, in the confocal microscopy, the material that we are using, the way we need some gases also continuously, and the amount that is needed in order to work continuously with the confocal microscope, it's in good amount. Every month it's take good charge of, or in Germany it was euros, in India it will be in lakhs maybe. And in order to, if something goes wrong a little bit while getting your image, any lens got break up or any things got messed up or electronics got messed up, um, and it's a very big, damage to the group, very big damage to the research that is going on. So we need to be very careful while we are working with sensitive any equipments. I mean, we are working with, if you're working with gel electrophoresis, it's okay. You're working with RT-PCR, it's okay. We're working with inverted microscope, it's okay. But sometimes things get yeah. But it's also depends upon from your one lab to lab. Once you are working in somewhere in a big lab where the lab has its own confocal microscope, then it's a no problem. Then you are go, because it's a lab, and the lab will be rich, and that lab might be the rich. So I got a chance to work in one lab actually. It's it's one of the richest lab in Germany. It's called Max Planck of Heart and Lung Research. It's in Bad Nauheim. It's a small, actually, village. It's a small village. Yeah, this is the place actually. You can see some images from outside. So this was the outside building. It was all lush green with all glasses. So we used to work on the first floor. The ground floor was for the Professor Didier Steiner, and the first floor was the for Professor Offermans, under which I was working. And inside it used to be like this. You just go from these stairs we used to go. On the first floor was our working place. This is the third floor, where the Professor Thomas Brown was the director of the group. So all group there was working on the cardiology, mainly in the cardiology. And our meeting used to be in these rooms. There used to be one presenter, and this was a presenter, and we used to have every weekly presentations. And and these are the all four professors there who are working. This is Professor Didier from USA. He switched to Germany. So he was our boss, big boss. He's a medical doctor, Professor Stephen Offermans. Yeah, and I'm not aware of them actually. So if you just Google them, these scientists, because this remember me from my old days. So I just want to share some theories that how exciting is Stephen Offermans. So he's also on Wikipedia. He's so famous. You see, he's also on Wikipedia. Born in 1964 in Berlin, eine Deutsche Medizin und Pharmakologie. Um, so his lab was one of the richest lab. They are working on the what you say GPCRs. If somebody is interested to work on GPCRs, one of the finest lab in the world to work on. But you need to work hard if you got the chance. If they see you have the dedication, if you can work with them, just write an email on this. Any of you who is looking for PhD wants to work uh, in the field of GPCRs, and he's one of the top scientists in the world. Maybe he will get the Nobel Prize winner in upcoming days because of his work. Every work that he's doing is coming in nature or in the cell or science, and it's beautiful actually. It's very beautiful to work under their guidance. So I did my master thesis under them for 1 year under the field of cancer metastasis, and my boss was Professor Sterlich, um, Boris Sterlich. Sterlich Boris. And believe me, as compared to, I don't want to be, what you say, racist, but these people were really nice in the science. They gave me um, really good place to understand the science, what is science is all about, and how to work with patience and good efforts, how to create your own experiments, how to think as a scientist, how to make things done for the world. So it's an excellent place, students. So these things that I have created with you for all these images, um, yeah, this he was the one that I worked for, and he's a Professor Stephen Oppermans, and there's a Nina, maybe Professor Nina somewhere. Oh, Nina is not there anymore. Yeah. So just wanted to share something from my past. So you can contact these person. Just write if you want to look for any positions for PhD or for master thesis or anything else. Just send application on this one. Contact. So sorry for going out of the topic, but maybe some of you might be inspired to do research in that foreign land and go for it and to really taste what the science is. In India, you can also do it, of course. IITs, JRF positions are there. You can go for SRF, PhDs, MSc, master thesis, everything is there. IISc, अपना Indian Institute of Sciences in Bangalore, our IITs in India, CSIR labs, they're also working pretty good. So I'm just sharing from my own perspective. Now, confocal laser scanning microscope. Let's focus on this. So this is a in this we use a laser light actually. Yeah, a laser light is produced on a plane of XY axis. That is like normal microscope. From the top, the laser scanner is there. Laser light is brought on the with the glass lights. The only light from the plane of focus reaches the detector, and the scanned images are digitally recorded. The images from consecutive focal planes can be recorded and we get a 3D image out of it. And for the laser we use krypton and argon. There are some cylinders are there which is keep giving krypton argon value there. Very sharp images are we getting in them, quite high sensitivities for the images, and can be used for fluorescence mode and reflective mode. And then no background is coming in them, so you're getting very sharp image, very distinct. Any cell signaling pathway that you're looking for, any what do you say, any molecule is going from one place to another, or any stress is happening, redox signaling is happening, or sumosylation is happening, or ubiquitination is happening in the cell, all these post translational modifications, dimer formation is happening, we can check very beautifully in these microscopes. So it has quite a good number of applications as so far as we have discussed the microscopy. So in this we can see these microtubules coming up very nicely. This is the nucleus part now, and these small thread-like structures coming on the monitor. So some images from the confocal. So in the left image that we are seeing is 13 optical sections of Trichomonas immunostained with antitubulin and fluorescein isothiocyanate. That is your FITS. Fluorescein isothiocyanate, that is your FITS. And we have stained them with the antitubulin and this FITS. And what we are seeing, these green structures consisting of your microtubules. So all these structures that we are seeing is microtubules here. So PF is your here posterior flagellum. We should use some different color maybe. Posterior flagellum. AF is your anterior flagellum. C is your costa here at the corner. And this red one is exostyle. This is A like this. So compare, and on the right hand side what you're seeing is bright field. In the left is a confocal image. On the same thing that we are seeing is here bright field of your Trichomonas, where we can see in the light everything is shining and we don't know what is we just get a very glimpse of the images, not particular nicely done in that. So we can see this exostyle. Yeah, we can see some posterior flagellum here with this green arrow, and the red arrow is showing the exostyle of that Trichomonas. But indeed, the image that we are getting in the confocal are far better. We can see how this structure is being making this digitalized, this wells here, very nice wells, and we can see all other cells making these structures of your Trichomonas. So that's how the difference between your bright field and confocal image is. Now we are done with your mainly your light microscopy. We have discussed quite number of microscopes so far. Now comes your scanning and transmission electron microscopy. Scanning and scanning electron microscopy and transmission electron microscopy. So scanning are the one in which first our specimens need to be dehydrated without any H2O molecules, and should be mounted over the they are called stubs are there. We will discuss what these stubs are, and the stubs and the surface of our samples are coated with gold or palladium or rhodium. Of our samples, the surface are coated with the gold, palladium or rhodium, and further they are dehydrated and mounted on these stubs. Then these specimens are further uh, placed in a vacuum chamber. Okay? After putting them in the vacuum chamber, an electron beam. So mainly, as we have discussed earlier case, confocal microscopy, it was a laser light from the argon, and difference between your SCM and TM is that we use a vacuum placed vacuum chamber, and we put electron beam on our sample, on the specimen, and these electron beams then scans and come back and forth over it. Like, this is our specimens, and this is our vacuum chamber, and it's there is a stub. This is your stub. Okay, this is your sample. Some sample here. Then you put your electron beam. It will come and go back somewhere, and it will go and come back somewhere. And so, as we have know, our surface is coated with our gold, palladium or rhodium. So these electrons beams that are bouncing back, they are collected and they are converted to an image. Yeah, they are converted to image and displayed on a TV like monitor. And in order to to handle your electron beam, we focus them with the help of magnetic field. So we keep one north pole and south pole. We keep a north pole and south pole. North pole and south pole, so that our magnetic field are going straight forwardly, they are not going here and there. So at the end, the result is that you get a 3D image. The same principle is actually used for X-ray crystallography. Let's say you have a car. This is a car, very nice car that we used to make in our fourth class, in our back pages. Just remember our old days. And we also used to put this exhaust in the end and making this exhaust coming out, a headlight, some doors, you know and then some handles, separation of a windshield. This is our car is complete. And I want to see that, I don't know, I'm blind, I don't see things in the world. But what will I do if I want to see something, whether it's a ball, whether it's a rectangle, whether it's a circle, I don't know the shape. I need to hold them in my hands if I'm not able to see, if I'm blind, right? So if the things are in my hands, then I can recognize the shape and can predict, okay, this is something round in my hand. And this method of scanning electron microscope is an indirect method. What we do, we don't have our things in our hand and we are able to see the picture. What we do, we get these laser lights, yeah, and they get deflected. Oh, sorry, electrons. These electrons coming, they hit here, and this electron come, they hit there, and this electron come and hit there. And we are getting all these details over the detector. This is my detector connecting to the computer, TV like monitor, computer. So this will keep giving me data, like if I put this, so this dot, it will be there, this dot will be there, then this dot will be there. So these dots will come together after hitting on this place. So I will keep hitting them, maybe from here, then going there, and then I will have eventually at the end a 3D image of my sample. That's basically how your X-ray crystallography is working. That's how your scanning electron microscopy is working. Yeah. So you get somehow, it predicted around 99% right structure at the end of your things. So it's give external topography of specimen. The topography means you can see from the top that how your structures looks like. and we get really high magnification. Sorry, I made these things here. High resolutions and high magnifications than possible in the light microscopy. So even we have discussed 7 types of microscopy in that, but this is the best one so far. And even we have discussed the confocal is giving the best, but scanning is even the farthest best of that. I don't know if in India how many labs we having. We have to check. But if we are having, we are doing good. So somehow I feel that we should, our government should spend some money into the research. But as population is so much high, our demands are different as compared to the developed country. Even though we wanted to invest in the research R&D, but we cannot make the common person then survive. So we need to have this balance also as per our economy is. So these are some images. As I said, this is scanning electron microscope images. So these top ones are with the SCM. Both are from the SCM, and the bottom ones are the DIC from the light microscopy. This is DIC, and this is your SCM. Sorry for my writing. And we can see particularly, even though we have seen some 3D images previously with the stained versions, but still there was a black background. In them, the images are so refined, so beautiful that each particles, each strain that what we are seeing one by one in a very refined way, which was not possible in couple of a few years ago. So both DIC microscope and SCM have revealed specimens surface topography. Much greater detail are obtained here using SCM because of the greater resolutions and magnifications capability. So what we're seeing here is Trichomonas, and the right hand side is your Trichohymena. And the bottom is also, this is Trichomonas, and this is your Trichohymena. So these are this is how we see in this DIC microscope, and the same image goes like that. So we can see the difference. The difference is uh, Zameen Asman Ka. I don't know. I'm remembering Hindi proverbs today, but I don't know how to spell them in English. Uh, Difference of Zameen Asman Ka. But I don't think so. We have any proverb for this. Difference of earth and air and atmosphere. Mm, yeah. So being fun apart, so we can see clearly SCM is far better than our previous light microscopy. So we are done with the SCM. Let's continue. As I started my class quite late, I hope no one has a problem if I continue for few more sessions, for further sessions. If somebody has a class for further at 8 o'clock, I think you have vaccine class maybe. Um, you can start with that, because we were told that we have to finish by it, but I need to finish this topic at least, so that our electron microscopy part is over by today. So transmission electron microscopy. So these, in this also we use fixed samples, dehydrated samples without water, and we, instead of using stubs, we embedded on them resins, hardened section stains with heavy metals. So we have a heavy metals as the bottom stable things. So we use uranium. I don't know. I'm forgetting English words today. It happens to me sometimes. Because the problem with me is the in the college where I'm teaching, all the students they speak Hindi, and for them it is very difficult to understand these Hindi things. Sorry, the English words there. So I need to keep remembering the Hindi words of each things, and this keeping me away from me English. And so I, it's not coming onto my tongue today. for some certain. That's the maybe the reason. But my fault. I will try to balance between our both Hindi and English, and that's an art actually. And then insert it into the electron column in the microscope. So electron beam is absorbed or deflected by the heavy metals stains, and shadows are cast into the film or phosphorant plate at the bottom of the column. So we get a not a 3D image in them, we got a 2D image. And but the interesting part is that so far the microscopy that we have seen, we were able to see the mainly the topography of the structure or the external structure, cell membrane, how the structure looks like from the outside. But this is transmission, as the name says, transmission. We are transmitting our electron into the structure, and because of that we are able to see internal structure of our cells with quite high resolutions, high magnifications, and again in this also electron beam is fixed by your focus by your magnetic field. So that's the comparisons between your SCM and TAM here. Are you showing you? I'm showing you at the moment. Trichomonas is the left one. Yeah, Trichomonas, and the so both are Trichomonas actually, but this is the SCM and the right one is the TEM. So we can see in SCM the tails are quite visible, but in this TEM, wow, everything is visible from the internal structures. It has shown us the nucleus, yeah. It has shown us the Golgi, yeah. It has shown us the hydrogenosomes. So this is some in Trichomonas. Somebody is going with the microbiology. For them, they might know. I'm not aware. What is this? Okay, they are equivalent to the mitochondria. It's written here. Then there is a flagellum here outside. Recombinant flagellum is also there. RF might be here somewhere written. Okay. Then cross sections of basal bodies, and further into the three anterior flagella. So you can see these very nice structures, interior and posterior. Um, I think this is also Golgi body here. No, they are not the Golgis. This is GC. I'm not aware, but we can see hydromonas, mitochondrial-like structures, Golgi, flagellum, nucleus, and many other. This straightforwardly these small, beautiful black dots, making this region to be covered. And with this was not possible to see these structures in the earlier cases of microscopy. So that's something you have to observe. This is, I always say, as a scientist, as a researcher, as a biotechnologist, or as a zoologist, or as a microbiologist, or as a genetist, or as a forensic scientist, whichever field you are, we are doing, we are just identifying the things which were not identified till now. So we are just identifying the unidentified things. So they are already present. So this structure was already existed. It's just it was not aware to the human consciousness, the human mind. We were not aware that there is a structure called Golgi. We were not aware there is a structure called nucleus. We were not aware there was a structure called hydromonas. And we were not aware that these structures look like this. And we were not aware that they have some corresponding functions. The nucleus has a particular functions, Golgi has a particular functions. We were not aware that whether they are present in this part and how this particular shapes looks like. So this is all, as a scientist, we have to think and explain to the public, which who have not identified till now from their own perspective, and those whoever scientists have worked on these fields, they have defined it, they have explained it, and all the world, and we are studying till now. So when we have started the first day with the cell biology, um, with the basic cell culture, it's we discussed all the structures there, but we were not aware that for how long they could have been, people have the scientists have taken time to identify them. So just think from that perspective also, think from the researcher perspective also, for how long that these things were still identified and then they were find out. There was also our Albert Einstein. I don't know, maybe 20 years ago or 70 years ago, he already told that there's a relationship between biology and physics, but nobody believes in him at that time. So he wrote everything in his paper about that. Okay, nobody believe me, it's fine, I will write what I know. So things, people who identified in the things in the far from the future, at that time they will, everyone will call them his silly. Same happens with the your father of genetics, um, Mendel. Yeah, Mendel's. So yeah. So more TM images, we can see rough endoplasmic reticulum here. Now, rough endoplasmic reticulum, some mitochondria is present, nucellus is present, yeah, nucleus is present, nucleus is present. So everything is well structured, nicely shown. These are the cristae of inside that they are showing here, and these are the rough endoplasmic reticulum is being present. And on the right hand side, we can see some zymosin granules now, and some cell boundary of these cells. And this is the images from your rat liver cells, and the right one is from the pancreatic acinar cells, revealing the internal structures. So that's the beautiful part of TEM. So this part we will correspond to the next time. That is your specimen preparation. So for all these three kinds of things, there are different way of doing it. I will take you to the different videos, how we do the microtomy, how we make the cell slides, how we make histology, how you make humoimmunostaining. It's a totally different world, and it's very tedious work also. I worked for almost 6 months for one of the PhD student. She was working on P2 Y2 knockout mouse in the Max Planck, and it was, yeah, it's a routine work, but you get really good results afterwards. That's the beauty. So okay, that's it for today. We will continue tomorrow. So we have just discussed today all about your scanning microscopy, and also we have discussed confocal microscopy and DIC image today. So we have discussed 1, 2, 3, 4, 5 microscopy today. So some of you might be having the classes for your um, vaccine technology. So go ahead with that class. Astronomical difference. That's the word I was looking. Thank you, Orishi. Mukherjee. Mukherjee is fine. Assignment you can submit, bache, whenever you are thinking that we have done with some topics. Just send me your things. I'm really happy to take. So some of students have sent me some messages on my Telegram. I was not able to reply because of my busy schedule since a few last few days. But I will try to reply them by today night, maybe after this lecture. Yeah. Okay then, take care till then, and we will have our classes this time on Saturday also, because we were not able to take class last week on Friday. So we will see us on Saturday also this time. Okay then, class, take care. We see us again um, tomorrow then. Until then, take care of your near and dear ones, and yeah, stay safe. Bye bye. Email is the best thing actually, not on the Telegram. Email will be better. Okay.